I'd like to just give more of a basic science presentation today, which will incorporate a little bit more information about the fundamental molecular and cellular defects that, as we understand them now, that may underlie AHC and also the clinical spectrum of disorders associated with other ATP1A3 mutations. And then I will summarize some of the work that we've been doing over the last 3 years with the generous support, and that includes our efforts to try to identify possibly new drugs or starting chemical compounds that specifically target the mutant ATP1A3 protein. The other discussion that will follow is a little bit of an introduction into some mouse models of ATP1A3 disease, one of which we've incorporated into our lab recently to begin to try do empirical short slang for saying we're going to be trying empirically drugs in these mice to see if we can't find something that we're or modify attacks. And I'll be doing this in a tag team with Dr. S. Our efforts to understand the fundamental dysfunction of neurons from human stem cell derived neuron populations that may give us greater insight into the disease biology, and also maybe another platform for testing candidate drugs or novel compounds. So the first part, again, I wanted to give kicked off very nicely some fundamentals about what is ATP1A3. What does it do, and how do we understand it in terms of its normal physiology? How do we then appreciate what goes wrong when there's a mutation in AHC, for example? First of all, ATP1A3 encodes a cell membrane ion pump. We hear the word channel sometimes and the word channelopathies, and I'm a purist, I apologize. Ion channels are one class of protein that allows ions, maybe sodium, potassium, chloride, calcium, variety of ions, to flow through a cell membrane. But pumps are very different. Channels can't move ions in a way to accumulate on one side or the other. They're very passive. Pumps have a lot of muscle. They're active pumps. They require energy from the cell, and they can generate very impressive gradients of ion concentration across the cell membrane. I'll say more about that in a minute. So this is just a cartoon of what might be a tiny little sliver of a cell. This is the outer boundary of the cell. Say this is a nerve cell, and the outer boundary of the cell, or the membrane of the cell, is made up of a pool of very greasy, slimy, oily, and all our cells have this plasma membrane lipid, and it is really impermeable to water. And I will learn this thing quickly. So anytime something has to move from outside of the cell to the inside of the cell, it has to get through this oily layer. So for things that are in water, dissolved ions, sodium, chloride, potassium, they can't just walk through this greasy layer. They have to have a pathway. Some pathways, like I said, are channels that just form a little con through the membrane through which sodium ions or potassium ions can just flow. And they flow because there's more ions on one side than the other, and they flow until the concentrations on either side are balanced, or you have equilibrium. Pumps, on the other hand, because they use energy from the cell, and what was illustrated, the energy comes from breaking apart this molecule called ATP, is like the gas of a cell. And it's combusted, and by breaking it apart, there's a little spark of metabolic energy that can drive things in the cell. And one of those things are these ion pumps, like ATP1A3. So what this thing does in a schematic form, here again we have this membrane illustrated by these funny things that are to represent membrane lipids in protein, which is just shown by these yellow shapes. And this is the pump, which by its mechanism derives energy by catalyzing a reaction that splits ATP into ADP and a phosphate. That energy is then transferred into the protein, which undergoes some gyration, and that gyration moves sodium across the membrane in one direction, from inside the cell to the outside, and at the same time, or subsequent, potassium to inside. Now, this is the point I always have to remind myself that most of you probably haven't had chemistry. Sodium, the abbreviation, the atomic symbol for sodium is Na. Makes sense, doesn't it? And potassium is K. And these are either Greek or Latin for natrium and kalium. I don't remember. Is it Latin or Greek? I don't remember. But so Na is sodium, potassium is K, and what this pump is doing constantly in every cell of our body. Now, ATP1A3 is only in nerve cells, but other cells in the body have different genes encoding very similar proteins. This is a pump that's constantly removing sodium from inside the cell and depleting cells of sodium. So the inside of cells has a tiny bit of sodium relative to the blood and the outside of the cell, which has a very high concentration of sodium. On the other hand, it actually loads the cell with potassium and depletes the outside environment, blood, extracellular fluid, of potassium. And then inside the cell is this massive amount of potassium. And so it's doing that so that there's this very gradient of sodium and potassium. And the reason it's doing that will become clear in a moment, but I'll just say that that's the battery. It's charging a battery that can be then unleashed to fire electrical impulses in the nervous system. So this is just a cartoon. This is a more modern depiction of the actual protein, and this is a protein derived from X-ray crystallography. You don't need to know more about that. And here's just, again, some fundamental biology of why we need to understand what these pumps are doing. First of all, if you looked electrically at a nerve, you stuck an electrode into the nerve and you recorded by some means what is the voltage in that nerve. What you would find is that nerve cells have an internal negative charge and relative to the outside. And the reason they have that is because of the action of the sodium-potassium ATPase. That's the formal name for the kind of protein that is encoded by the ATP1A3 gene, or these ion pumps that I described. And again, it's removing sodium from inside and partially replacing it with potassium. And so what's illustrated here very importantly is every cycle of the pump ions out and 2 potassiums in. It's not balanced. More potassium, more sodium is removed from the inside of the cell than is replaced by potassium. And because each of these ions has a single positive charge, you end up with an imbalance of charge. And just to illustrate that a little differently, a little interpretive dance by Drake to help us. So again, it's the stoichiometry, as we say, is 3 sodiums for 2 potassiums, and 1 ATP molecule bites the dust. Anyway, beyond this hip-hop illustration, one can, again, conceptualize what happens when you have this charge imbalance because of the ATP1A3 or any sodium-potassium ATPase. So this is a cartoon again where there is some conditions on one side of this membrane. This line just represents a cell membrane, for example. and there's something, a pathway in between. And let's say, for example, the right side is the inside of the cell, and the left is the outside of the cell. And when there's a charge imbalance, and we can monitor that with a voltmeter, cartoon above, and you just have a charge difference of one positive charge, the inside of the cell, becomes negative relative to the outside of the cell. When you look at a nerve, the inside has a negative charge. And that negative charge is reflected in what we call the resting membrane potential. I guess I put a slide in and didn't show you. I'm sorry. The resting membrane potential is what we can measure directly in a live cell as evidence that the pump is working. Now, there are other things that factor into the resting membrane potential, and I'm going to just simplify it by not telling you what those are. But ATP1A3 is the most important one to do the initial charging of that membrane. And secondly, it's most important for separating sodium and potassium to create this sort of potential electrical energy in the cell. And the way the cell unleashes that potential electrical energy is it opens selectively channels, not pumps, but channels for sodium and potassium. But it does so in a stepwise fashion, so that one opens a sodium channel, and suddenly all that sodium that's been outside the cell can get in and rushes into the cell. Now you've got this flood of positive charges rushing into the cell, and that previous negative internal charge of the neuron just goes away. Suddenly, in a localized environment where those sodium channels are opening, there's this sudden jump in the voltage in that part of the cell. And instead of being negative, it becomes very positive. That only lasts about a millisecond, because the cell then turns those sodium channels off and opens potassium channels. And now, because potassium is enriched inside the cell, potassium then flees the cell through that potassium channel, and the membrane potential does the opposite. It goes back to being negative. And so this sort of funny balance between sodium and potassium is critical for what is called the action potential. The action potential is what we show in nerves that are undergoing stimulation. And so if we stimulated a nerve, we would see this rising and falling of the voltage across that cell. And this brief pulse of electrical change can then travel down the nerve. This is essentially the electrical signal that our nerves conduct. So when a signal is started in one part, sorry, those are ugly, aren't they? It can actually propagate along the nerve until it reaches the end of the nerve, where it can then do a secondary signal. And so this is the fundamental reason why ATP1A3 is so critical. It establishes the internal negativity, the negative charge of the cell. It separates sodium and potassium as if it were charging a battery. And that the electrical juice that allows nerves to have electrical signaling in the form of what we call action potentials. The reason I'm showing you here, this is a big squid, is because we know these things because squid has a gigantic nerve. It has what's called a squid, it has a giant axon, which is a long process tied to a nerve. And 70 years ago, a couple of guys named Hodgkin and Huxley exploited this preparation to figure out everything we know now about how nerves signal electrically. And so again, another illustration of an action potential comes from this old crusty model, whereas if you can stimulate this long nerve, this thing that looks like a sausage is the nerve, you can stimulate it here on the left but then record at different time points, A and B, to the right. And what you find is that at point A, after the stimulus, there's this rising and falling of the membrane voltage and action potential, and then it travels, and you see it again, but further down. So this is evidence of this fundamental electrical signal called the action potential and its ability to travel along the nerve. And again, none of this would happen if there wasn't this charging of the battery with the sodium-potassium ATPase. Now, I know that's a lot to handle, and I'm happy to clarify that in some Q&A. But let me just go on and tell you a little bit about some of the things we've been working on over the last few years. And I'll start with one of our primary goals has been to examine whether it's possible to identify compounds or drugs that can directly act on ATP1A3. There isn't one. There are things that inhibit ATP1A3, lots of them. But there's nothing that really activates it that's been demonstrated. So one of our goals was to look at that and see if we could come up with a strategy to find activators of the pump, because that would hypothetically be beneficial in situations where there was a loss of pump activity, which we think is the reason for most the underlying molecular dysfunction responsible for most of the ATP1A3 mutations. We think we need to do this not only in a laboratory setting in a cell system, but also in animals. So I will introduce you to some new friends of ours, an animal model that what was shared with us recently from Aarhus University in Denmark. And then lastly, our work that I'll actually postpone from my talk and add on to the end of Dr. S's talk, which is our investigations of human neurons from patients with AHC to try to understand more of the cellular defect underlying this. So we need to understand the molecular defect, the cellular defect, and ultimately pull it into animals to do more serious investigation. So I just want to summarize for you what really was well over 2.5 years of work to try to find if it was going to be easy or hard to find something that would activate ATP1A3. So we've done that in 2 ways. And this is work that began when I was still at Vanderbilt University in collaboration with Kevin S. And has continued since my move to Northwestern. While at Vanderbilt, we took advantage of a regional expertise in measuring potassium channel activity in cells with a peculiar method called the Thallium Flux assay. I'll tell you about that in a moment. We adopted this for use with ATP1A3, and it worked. And we were able to at least learn something about the functional abnormalities in 3 of the most common ATP1A3 mutations, and also use this as a screening platform for testing several libraries of approved drugs and different chemical compounds to see if anything activated the pump. Since moving to Northwestern, I took advantage of another resource that was uniquely there, which was a very talented computational chemist who used computer models to do this kind of screening. And I'll show you the findings that we made there. Let me say at the onset that drug development is not a short-term process. It's a long-term process, and there are many starts and stops, and unfortunately many. The Thallium Flux assay is illustrated here. Thallium, by the way, is just another atom, like potassium, has about a similar size, and most proteins that can interact and transport potassium can also transport thallium. And we discovered, in fact, that the ATP1A3 pump could, in fact, pump thallium quite well. So we actually developed with the folks at Vanderbilt I mentioned before. The way this works is there's a fluorescent dye that is placed inside the cell. These little green dots are a dye. When they come in contact with thallium ions, that are gaining entry into the cell through some pathway like the sodium-potassium ATPase, they ignite the fluorescence of this dye, and you can read the fluorescence and quantify it and make measurements. And so this is an example of how that worked. Not a neuron, but a cell that had ATP1A3. We then exposed that cell to the dye and made sure that the cell incorporated the dye. And then we exposed the cell to a solution with thallium ions, with the idea being that thallium would then be pumped into the cell by ATP1A3, and the thallium would ignite the fluorescence of this dye. The fluorescence of the cells would start glowing, and we could measure the cells as they glowed and monitor it over time. And what we expected to see was that there would be a cumulative signal from the cells over time that would be proportional to the amount of thallium pumped into the cell. And so here we show the time from left to right of a signal we got from cells that had the normal ATP1A3. WT in this slide stands for wild type. An old genetic term, it means the normal. And so what we see is a very rapid rise and a plateau of fluorescence, what we interpreted as thallium uptake, showing that this could be used as a way of monitoring activity of the ATP1A3. Now, what happens if the cells, instead of the wild type ATP3, had a mutation? Well, here's one experiment where we looked at the three most common mutations, D801N, E815K, and G947R. And all of them have much less activity than wild type, or what we would label as loss of function. Some seem worse than others, but there's a caveat in this experiment, which is these are cell lines that were selected for the amount of protein they expressed, so we can't really directly compare them that much, but at least we know that these qualitatively show loss of function. And so therefore, again, the rationale was, can we find something that would rescue these by activating the pump in some way? So using the thallium, Christine Simmons, who's here at the meeting, at Vanderbilt, screened nearly 10,000 drugs and compounds. This included 2 independent libraries of all approved drugs, either FDA or EMA approved drugs, and about a little more than 6,000 or almost 7,000 other compounds that were really represented a diverse array of chemical compounds that were thought to interact with ion channels or ion transporters. This is just some images from the assay, but I will just tell you that there were lots of what we call hits, lots of positive signals, things that increased the fluorescence of the cells taking up thallium, even though they had the mutation. But when we went back and looked at some secondary analyses to rule out artifacts of the experiment, which is always done, we found that virtually all of these, maybe with the exception of one, were what we call false positives. They weren't really true signals. And so eliminating those, we end up with really out of 10,000, it might have been one that we still are not 100% sure about. So this is an ongoing concern. We think this assay is still very valuable, but the background cell type has to be adjusted so that we don't have such a false positive rate. As another strategy, again, when I moved to Northwestern, I was introduced to this very talented computational chemist named Rama Misra, and he has a specific talent. He can model a protein in his computer based on structural data from the literature, and then dock individual chemical compounds one by one into spots in the protein that look like drug binding sites. And he can actually do this for 10 million chemical structures. So whereas we spent probably a better part of 2 years screening 10,000 compounds, took him about a week to screen. But it's all theoretical. That said, so this is the model of ATP1A3, illustrated a little differently than before. He noticed, and he has some computer programs that scan the surface of these molecules, looking at every little crevice and crack in the protein. And I said, look in the area around aspartic acid 801, D801, most common mutation in AHC. Look in that region and tell me if you see any binding pockets. And he said, yeah, there it is. And in fact, there was this little spot. He said, like, that's a binding pocket. We could assay that pocket for compounds to bind. So he did that. So he docked 10 million in his computer. And that came back with 57. 57 of the 10 million seemingly and stably into that pocket. And you might think, well, that's terrible, 57 out of 10 million. That's about right. He said the average for this type of experiment, because they do it under highly stringent conditions, is about 70, right on target. But that's just step one. This is the discovery part. The second part is we needed to come up with a way of asking the question, do these rescue an ATP1A3 mutation? Does it have any effect? So we actually, just another illustration showing, again, one potential compound and how it fit partially into this binding pocket. And then he can even use even a more high resolution molecular imagery to show exactly how it fits and how it's predicted to interact. So a little more above my pay grade. So again, we turned back to a biological assay using cells that expressed the known ATP1A3 mutations. And we took advantage of the fact that if we introduced a second mutation remote from the disease-causing mutation, we could make the cells survive in the presence of an ATP1A3 inhibitor known as wabane. Wabane is a generic inhibitor of sodium-potassium ATPases, and it kills cells, unless somehow those cells have an ATPase that withstands the effect of wabane. On the left here are 4 little wells of cells that have been grown up and then stained blue, so you could see them. The upper 2 wells, which are marked by WT, have been exposed to either the normal solution, called control solution, or enough wabane to kill them. And because we've engineered this wabane insensitivity or wabane resistance into the cells live, and they remain blue. D801N and all the other common ATP1A3 mutations. D801N, for example, when the cells expressing that mutation are grown in the normal media for the cells, they live just fine. As at wabane, they die. And the reason for that is the wabane kills the cell because there is no functional PAse that is been rendered resistant to wabane. So this is a very crude but effective cell survival assay. If cells live in the presence of a compound, for example, the compound has activated or rescued the mutation in some way. And so this is an illustration of a more kind of quantitative view of that. Again, work done by Christine Simmons. And that what happens if you add progressively more and more wabane. The horizontal axis of this graph on the right illustrates the concentration of wabane. And when you get out to 1, 2, and 3, you're talking very heavy doses of wabane here. And been rendered wabane insensitive, which are the red line. They're fine. Wabane doesn't bother them at all. But these mutations associated with either the cells that are just garden variety cells, HEC293 cells, or cells that express a couple of the mutations, they actually die off very effectively. So we can exploit this to assay these compounds to see if any of them work. So Christine checked all 57 of them in duplicate. I'm not getting the slider go. And what you came up with, there were 5 of the 57 that rescued substantially the cell survival in D801N expressing cells. And so this, again, some raw data. It's a little hard to understand, but the lines at the bottom represent the amount of survival in cells that were treated with Wabane that don't have any ATP1A3, and basically none of them survive in Wabane. Above here are cells that have the wild-type Wabane-resistant form of ATP1A3, and they all live. And then in the middle here are mutant cells, cells expressing D801N with a Wabane resistance mutation that have been pre-treated with some of the compounds. And unlike most of the 57 compounds, which didn't do very much, these 5 prevented cell death substantially, about 50%. And so these are still under a lot of investigation in our lab. We're looking at these very intensely now to try to determine what are these real? Are they having an effect on ATP1A3? Can we learn anything about the divergent chemical structures in some way to try to build a case for maybe we have an activator? So we're not quite there yet, but this is just a fundamental story. These, by the way, are compounds that don't look anything like a drug. They are random compounds from a chemical library. But the good news is we have very talented medicinal chemists at my institution that can look at them and start to play around with them chemically to see if they can't engineer in properties that are more drug-like and also maybe intensify the activity that we see in this assay. So that's kind of update. Moving on, and I'll try to make the next few slides pretty brief. We have done a couple of things in the last year to try to import mouse models of ATP1A3 related diseases into our laboratory. This is just a list of all existing or known mouse models, at least those that have been published or at least disclosed in meetings. We know the work of Steve Clapcoat in England. He's developed or cultivated and characterized the Michigan mouse, which has an ATP1A3 mutation that arose from a chemically induced mutation. It wasn't a human mutation, although subsequently I think the same mutation has been identified in a child in China, so it is a relevant mutation. Dr. McCarty at Duke and his team have made the D801N mutation in a mouse model and have published a beautiful study a couple of years ago illustrating its properties and how close the human clinical features of AHC. His lab is also, I think, working on publishing their mouse K. A group in Denmark at Aarhus University, it's just a lab of Karen Leakey-Hartman and her postdoc at the time, Thomas Holm. They've actually submitted a paper looking at mice that have an engineered mutation that was found originally in a patient with RDP, but subsequently in other subjects, some of whom have more typical symptoms of AHC. But this mouse model, and I'll illustrate a little bit of that for you, has been presented at the ATP1A3 conference last year in Bethesda, and it mostly has features of AHC. It does not have the permanent dystonia that one sees in RDP, and it really looks for all the world like an AHC mouse model. Lastly, my lab at Northwestern has been engineering the third most common mutation, G947R, in a mouse model. These mice are being bred now and being genetically tested, so we're not 100% sure we have the colony growing yet, but we're making really, really good progress. Thomas Holm, who was the postdoc in Karen Hartman's lab in Denmark, who helped characterize the D801Y mouse, is now in my lab at Northwestern, and along with him came the mice. Not on the same flight. We didn't want to get him arrested the first time he came to the U.S. So anyway, this mouse model has a number of features that are reminiscent of AHC, including stress-induced attacks stress-induced attacks of weakness and dystonia. They also have a pretty prominent ataxia that's supposed to be illustrated here. I tried to make that happen automatically. There it goes. So the mice above are normal. The ones on the bottom really have a lot of trouble. They really have a very unstable gait. And really lose the race very effectively here. The other thing that you can do is you can place the animals in, that may start, there we go, in some cold water. Cold water exposure is enough of a stress that they reliably attacks of both dystonia and hemiplegia. So the two mice on the left are normal mice, and they're just shivnits. They get up and move. The other two mice here are, and there's a little bit better explanation here, or little better illustration here of an animal that's has right-sided hemiplegia. Is working hard to right the mouse, but the right side is just completely flaccid in this case. And so we think this is a very quantifiable trait that could be used to test the potential efficacy of different approved drugs, for example. I think there was another picture here, and this is actually just another similar experiment where the animals were for a few minutes. The difference in this experiment, in the middle are the north flanks. To the left are mutant mice that have not been treated, and on the right are mutant mice that have been given an injection of minutes before their, this is just a shivering over here violently. This weakness. The other animal is really just sort of laying there, much about it. But 30 minutes or so, the normal animals are over here, kind of having a meeting, kissing and grooming themselves. And, they're fine, they're okay. The untreated animal back here is still having this weird tail movement. I think it's going to roll around here in a minute. Actually is still pretty weak, whereas the flunarizine mouse didn't look as bad. So we're presented at the Bethesda meeting last year, and I don't take credit for them, but Thomas is. So those mice are breeding very well. We're hoping to develop and can actually expose serially to different. We have a long list I'd like to try. Ultimately, we hope that some of the work we're using the computational chemistry for may result in some testable compounds as well. So having that animal platform is really key. And so just the last thing is, and this is going to be a bridge to Dr. S's talk, where we've collaborated with Dr. S's lab for the last 3 years or so to really get to the point where we can study human neurons from AHC patients. And this is exploiting the technology that he will tell you about, which is to use induced pluripotent stem cells, or so-called iPSC cells, to be able to generate nerve-like cells in the laboratory, essentially create a disease in a dish, if you will. In our hands, we're using of an approach using electrophysiology to investigate what are the fundamental properties of those neurons. And so I'm going to show you a little bit more data after Kevin's introduction and his talk. And so with that, I'm here, including currently in my lab, or I think there's a name missing, but Sorry, I won't touch anything anymore. But I mentioned before, Christine Simmons, who's here at the meeting, did all the in vivo screening. She's also been very involved in the iPSL studies of late. Thomas Holm, who's moved from Denmark to Chicago and has been very helpful in instituting the use of the D801 MOS, and our other important collaborators, and of course funding from the AHC Foundation, which made all of this possible. So thank you very much. So I'm happy to take a few questions before we move on.
Thank you very much. I realize that the more I learn about this, the less I feel I really know. And so there's a simple question, which is, when you showed the mice with the flunarizine, you had preceded that with the idea that there were 57 hits and the no hits. What does flunarizine have that might relate to the 57, or what do we know about flunarizine, why it works?
Flunarizine is not a very specific drug, and there's a lot of potential targets, and there's a lot we don't know about it. Structurally, it's probably unrelated to any of the hits that I showed, because flunarizine is not acting on ATP1A3 directly, as far as we know. It's doing something indirectly on the neural circuit that somehow stabilizes the condition, and that's probably going to ultimately be how this disease is most effectively treated anyway. But there's still hope that maybe knowing what the mutation is, what's the mutant protein, if you could target that in a specific, has been done in cystic fibrosis, for example, and maybe in some other diseases. But flunarizine is probably unrelated. Its effect is probably unrelated to a direct, as far as we know.
Thank you, and thanks for all the work, too.
Pleasure. Question in the very back. Oh, sorry. Yeah.
I just wanted to mention, I've put my kid in warm water, 95-degree pool, and he still got paralyzed. So, I mean, the fact that some of the mice were shivering, I don't know if the water has to be that cold to do the same thing. He will not get paralyzed in a bathtub.
In hot water.
Yeah,
in hot water.
But the pool was at 95. Which is pretty hot, and
yeah, that's very hot pool water.
Right. Yeah, my sister's husband wanted to.
Sounds like a jacuzzi.
No, it was a large pool, and he was fine. I was just wondering if you will try that dependent.
We are very much interested in the temperature-dependent phenomenon. We in fact are set up to look at hyperthermia or high temperature, which is an assay we've been doing for years to evoke not these. So we will actually try that. We'll see if fever evokes an attack, but or elevated body temperature. But you know, again, there are a lot of things that could be stressful about entering a pool. A young child entering a pool, that might be a frightening experience. So it might not be just temperature in that setting.
He loves it. He wants to run into them.
Okay.
Yeah.
Question regarding one, thank you for all the fantastic work you guys are doing to advance the knowledge here. I guess a question in looking at kind of the science overview and then looking at some of the triggers and helping to understand how the triggers model. I mean, is it kind of like a rapid depletion that causes, if you think of triggering activities and what you were talking about, how the sodium pump works, is any insight into the relationship?
It's a great question, a really great question, and into that. Certainly, we were just talking about temperature for a moment. You know, certainly temperature affects lots of things, and certainly it will affect the sodium potassium pump that we were talking about. Is going to work slower at lower temperatures, but the normal ones too. So what? Maybe there is some critical threshold that you're breaching when you go below a certain temperature. That's speculation. Other stressors, I don't think we have a great idea of what those are. If I had to speculate, I would say there they're probably acting at a more of a neural circuit level. But honestly, I don't think we know. It's always, in many neurological diseases that have paroxysmal symptoms, the triggers are usually the last thing we understand.
And I'm sorry, just one quick follow-up question. If you looked at the kids when they're in an episode versus not in an episode, would the electrical activity look different?
Someone who examines kids would have to answer that. You don't want me to answer that. Maybe Kevin.
Dr. S?
He said he thinks so.
Thank you.